So hello guys, we will be starting our new lecture on electron transport. Okay, on we will be first starting with electron transport. Okay, so what is electron transport? Electron transport chain. So what is the electron transport chain? We have to first understand that. So basically, electron transport chain is a series of electron carriers over which electron pass in a downhill journey releasing energy at every step, that is used in generating an electrochemical proton electrochemical proton gradient, which helps in synthesizing ATP-like compounds. Okay, so what is the electron transport chain? Electron transport chain is a series of electron carriers. Suppose cytochrome, ferredoxin, these are some electron carriers which is present. I will be talking about it later. These electron carriers carry electrons in a downhill manner. Downhill in which sense? Downhill in redox potential level. Okay, so this statement is very important, that the journey of the electron is a downhill journey. This is an important, very important information here. While the electrons are journeying in a downhill fashion, they are continuously releasing energy at every step. Okay, so thus while the electrons are passing through an electron carriers, they are going in a downhill journey and they are releasing energy like ATP, like they are releasing energy at every step, and that energy is used in generating an electrochemical proton gradient. That means this flow of electrons helps in accumulating proton in a particular site, that is under the lumen of the thylakoid. We will be talking about it later when we will be talking about chemiosmotic hypothesis. But the protons accumulate in the thylakoid, and outside the thylakoid there is less proton, so there is a proton gradient. Okay, there is a proton gradient, and this proton gradient between out of the thylakoid, and in the thylakoid there is a proton gradient, and this proton gradient helps in releasing ATP like compound or synthesizing ATP like compound. So this is a electron transport chain. Okay, so the important statement I am noting it down. So electron transport chain always functions from high redox to low redox. Okay, to low redox potential. This is an important statement. So basically there are two types of electron transport system, two types of electron transport system or chain, whatever you call it. Electron transport chain, there are two types. The first type is non-cyclic and the second type is cyclic. Okay, so first we will be studying about the non-cyclic electron transport chain, or you may call it also photophosphorylation. So electron transport chain or photophosphorylation is basically two types. There are various books. In various books, they are written in various concepts. So basically, these are two types. First is non-cyclic, and the second is cyclic. Okay. So we'll be first starting about from non-cyclic photophosphorylation. Okay. So first we'll be starting from non-cyclic photophosphorylation. Okay, non-cyclic photophosphorylation. Non-cyclic photophosphorylation. So in non-cyclic photophosphorylation, there is, first you have to understand. Okay, I am taking it to the next sheet. Non-cyclic photophosphorylation. So basically you have to understand there is a source of light which is sun. Okay, sun is radiating its lights of various wavelengths in various directions. Okay, there are some lights which is incident on LHC. Remember the light harvesting complex, and there are some light incidenting on chlorophyll A, which is a reaction center, and this LHC here passes its energy to chlorophyll A or reaction center. Okay, and this photosystem here is photosystem 2, which happens to be functionally most active in 680 nanometer, photosystem 2. Okay, and this reaction center or this chlorophyll A passes its electron passes. Okay, so first there is sun, and there is incident energy from to photosystem 2, and this energy here helps in splitting the water into two compounds, okay, into various components. This energy helps in water into splitting in H2O is equal to 2 electron plus 2H plus O. Okay, this O is released as a byproduct as oxygen, remember, and this electron here enters this pathway here, and this electron passes to an electron acceptor here, electron acceptor here. So redox potential, so this electron acceptor has much lower redox potential. This electron acceptor has much lower redox potential, than photosystem 2. Okay, then photosystem 2, okay, and this electron acceptor passes its electron to various electron carriers such as plastoquinone. First there is plastoquinone, I am sorting right in PQ, plastoquinone. Then there comes cytochrome, then cytochrome passes its electron to plastocyanin, I am writing it PC, plastocyanin, okay, PC, plastocyanin. Then this plastocyanin transfers its electron to the light harvesting complex of photosystem 1. Okay, and you have to remember a special thing, that this sun here is also radiating electrons, radiating its wavelength to photosystem 1 also. Okay, so the sun is also functioning in photosystem 1 also, in this area also. Okay, so first LHC of photosystem 1, then chlorophyll A is reaction center. In both chlorophyll A and light harvesting complex of photosystem 1 is receiving the sunlight, and remember photosystem 1 is functioning optimally at 700 nanometer of wavelength of light, and this photosystem 1 passes its electron to another electron acceptor here, which is also lower redox potential, sorry, I have a mistake here. The redox potential is high redox potential than photosystem 2, and electron acceptor of photosystem 1 is also at high redox potential than photosystem 1. The redox potential of these electron carriers are lower successively at every step. That means the plastoquinone has a higher redox potential than cytochrome, and the cytochrome have higher redox potential than plastocyanin. Okay, so the electron carriers passes its electron down a redox potential gradient, and this electron acceptor is at high redox potential gradient than photosystem 2. Okay, so this is the concept, electron acceptor. So basically there is an electron acceptor, and this electron acceptor passes its electrons passes its electron, and here comes the NADP, and NADP on receiving electrons reduces to NADPH. Okay, this is NADP plus, and this is reduces to NADPH. So you have read that oxidation is loss of electron and reduction is gain of electron. Okay, so NADP plus is gaining electron, so this is the NADP plus is getting reduced, and on getting reduced it is forming NADPH. Okay, so this is very simple, and you have to remember these electron carriers here, while passing the electron, converts a ADP to ATP. These electron carriers, while passing these electrons, converts a ADP into ATP. Okay, so basically what we have learned, I am summarizing. The sun, I am taking a highlighter. The sun is radiating its wavelength to the photosystem 2. Okay, photosystem 2. There is chlorophyll A and in light harvesting complex, whatever, and the chlorophyll light harvesting complex is supplying energy to chlorophyll A, and the chlorophyll A, this energy which is accumulated in photosystem 2 helps in breakdowning of water or photolysis of water. This is called photolysis of water. On H2 on photolysing gets 2 electron plus 2H plus 1 oxygen atom, and this chlorophyll A passes electron to the electron acceptor. This electron acceptor is at a higher potential redox potential gradient than the photosystem 2, and this electron acceptor passes its electron to various electron carriers, namely first is plastoquinone, then is plastocyan, then first is plastoquinone, then is cytochrome, then is plastocyanin. Okay, and these electron carriers are arranged in a down potential gradient. Okay, so while passing the electron, first the electron comes with plastoquinone, which has a higher redox potential than cytochrome, and from cytochrome the electron passes to plastocyanin. Then cytochrome has a higher potential gradient than plastocyanin, so successively the redox potential is getting low. Okay, and this plastocyanin transfers electron to the photosystem 1, and this photosystem 1 again pumps the electron to the electron acceptor, which happens to be at a higher redox potential than photosystem 1, and this electron acceptor passes the electron to other electron carriers, and through this electron carrier the electrons are accepted by NADP plus, and NADP plus gets reduced to NADPH, because oxidation is loss of electron and reduction is gain of electron. NADP plus gains electron, so NADP plus gets reduced to NADPH. Okay, so this is the basic concept here, non-cyclic photophosphorylation. And look here, interesting thing, that is the interesting thing here is the electrons comes at photosystem 1, then it goes up, then it again goes down at a at a electron at a level of redox potential gradient. At a level of redox potential gradient, it first goes up, then it goes down at a redox potential gradient, then it again goes up at a redox potential gradient. Okay, so basically what you are saying, what you are seeing, this shape is forming, and this shape is a inverted jet, inverted Z. Okay, so non-cyclic photophosphorylation for this reason also called as Z scheme. Z scheme, okay, Z scheme, SCHME, SCHME scheme, Z scheme. Okay, I am not that good in alphabets or the vocabulary. Let's correct it. So this Z scheme. For this is also called Z scheme. And the next comes our next thing is about a conceptual clarity that we will be talking about. Okay, so basically, so suppose this is a thylakoid. This is a thylakoid outer membrane, and this is a thylakoid inner membrane. Okay, so basically what I am trying to say is this photosystem, this photosystem 2 is inside the thylakoid. Sorry, I've drawn it wrong. Sorry. There is a one membrane in thylakoid. Sorry, correct it. There is a one membrane in thylakoid. This is thylakoid. So this photosystem 2 here is inside the thylakoid membrane, and the photosystem 1 is outside the thylakoid membrane. Okay. Why I am saying it? Because while the H2 splits here in the photosystem 2, while the H2 splits here, H2 gets converted into 2 electron plus 2H plus a nascent oxygen. This nascent oxygen 2H plus and 2 electron are formed inside the thylakoid. Okay, and while NADH plus get reduced to NADPH, it forms at outside the thylakoid membrane. Okay, this reaction, because this reaction is associated with photosystem 1, and this reaction is associated with photosystem 2, so it happens under the inside the thylakoid membrane, and this reaction associated with photosystem 1, so it happens outside the thylakoid membrane. It is a very important concept. Remember this. Okay, and an important thing about Z at non-cyclic photophosphorylation is in non-cyclic phosphorylation continuous supply of electrons are required. Why continuous supply of electrons are required? Because this NADP plus here get reduced to NADPH, and to get reduced electrons are required here. So because of this requirement of electron, the continuous supply of the electrons are required in case of non-cyclic photophosphorylation. So here H2 splitting happens continuously. H2O splitting happens continuously. Okay. So then we will be covering about cyclic photophosphorylation. So in cyclic photophosphorylation, the cyclic photophosphorylation involves only photosystem 1 photosystem 1 involves only photosystem 1 or which is active in 700 nanometer. Okay, and this cyclic photophosphorylation happens in stroma lamellae only. Why? Because I have told you previously, this stroma lamellae is the connecting link between the two granules. Okay, so this in this stroma lamellae the cyclic photophosphorylation occurs, and the non-cyclic photophosphorylation which I was talking about, the non-cyclic photophosphorylation which I was talking about happens in the grana membrane. Okay, happens in the grana membrane. Okay, understood? So happens in stroma lamellae only, and here no continuous generation of electron is required. Okay, no continuous generation of electron is required. So basically what happens, I am telling you in short. This is sun. The sun is emitting its wavelength to the photosystem 1. Okay, and the photosystem 1 pumps its electron to the electron acceptor, which is at a higher redox potential than photosystem 1. I have talked it previously, and it is like this. And this electron acceptor pumps the electron to the electron transporters, or to the ETS, or electron transport system, or chain, whatever you may call it. And this ETS then pumps back this electron again to the photosystem 1. And while this ETS pumps these electrons to the photosystem 1, this ETS converts a ADP to ATP. Okay, so this is a short cyclic photophosphorylation. Here you are seeing that clearly that no continuous generation of electron is required, because the electrons are flowing here in cyclically. Okay, and here is the important thing is only ATP is formed, no NADPH, only ATP. Okay, this is an important point here. So in the next slide we will be comparing the cyclic and the non-cyclic photophosphorylation. Cyclic and non-cyclic photophosphorylation. And at the first point, the cyclic photophosphorylation depends on PS1 only, and the non-cyclic photophosphorylation on PS1 and PS2 both. Okay, and in cyclic photophosphorylation, external electron source not required, because the electrons are flowing here cyclically, so no external source of electron is required. And in case of non-cyclic photophosphorylation, external source of electron is required. because the electrons are here consumed by NADP plus NADP plus. So here continuous electron flow is needed. So external source of electron is here required for non-cyclic photophosphorylation. Okay. And third topic is H2O. Here photolysis or the splitting of H2O does not occur, and in case of non-cyclic photolysis does occur. Okay. And here in case of cyclic, ATP is formed and NADPH is not formed, and in case of non-cyclic photophosphorylation, ATP is formed and NADPH is also formed. And in case of cyclic photophosphorylation, the reaction occurs at stroma lamellae, and in case of non-cyclic photophosphorylation, it happens in grana lamellae, or we may call it thylakoid, whatever you may call it, thylakoid membrane, sorry, whatever you may call it. Okay, because the thylakoid are stacked to form grana, and the grana is made up of thylakoid, so both are totally almost synonymous in case of this. Okay, so this is the main points between non-cyclic and cyclic photophosphorylation, which we have to remember. Okay, so in the next slide I will be covering the first light reaction. I will be covering the chemoosmotic hypothesis in the next lecture. We will be understanding it very in a little bit detail. Okay, because it is a very hard concept. We have to understand it in details. We have to go some deeper into the details to understand the chemoosmotic hypothesis. So I am starting from the biosynthetic phase here. I will be covering the next. I will be covering the chemoosmotic hypothesis in the next lecture. Okay. So this is biosynthetic phase, and this biosynthetic phase is also called as light reaction. Why this is called as light reaction? Because biosynthetic phase, this is not light reaction. Biosynthetic phase is called dark reaction. In some books this is called as dark reaction, and the biosynthetic phase is a phase in which the products of the light reaction or the photochemical phase is being used. So in some books this is written as dark reaction, but this dark reaction is not synonymous with biosynthetic phase. This is not a name of biosynthetic phase, because dark reaction means, so listen, because dark reaction means it happens only in dark, but the biosynthetic phase can happen also in light and in dark. The biosynthetic phase happens also in presence of light or absence of light. So you are seeing it, the dark reaction. So this is not synonymous with dark reaction, because it happens in presence of light or when light is not present, in both. Okay, whereas the light reaction occurs only and only when the light is present, but the biosynthetic phase can occur independently of light, because it requires the products of the light reaction, not dark. Okay, dark is not mandatory here. So dark reaction is a misnomer. Dark reaction is a misnomer of biosynthetic synthetic phase. Why? Misnomer means not same. Okay, the target and biosynthetic is not same. Okay, and in the biosynthetic phase, the products of light reaction or light reactions or photochemical phase is used. Okay, and what is the product of photochemical phase? That is ATP and NADPH and NADPH, which I have talked about previously. Okay, okay. So we will be starting about the first biosynthetic phase. We will be starting first point of biosynthetic phase, or the first thing to understand biosynthetic phase, that is Kelvin cycle. That is Kelvin cycle. Okay, Kelvin cycle. Kelvin cycle was first discovered by Melvin, scientist named Melvin Kelvin, a scientist named Melvin Kelvin. Okay, and Kelvin cycle is known as also C3 cycle. Okay, so Melvin Kelvin basically first deduced this cycle, and Melvin Kelvin deduced this cycle by using a radioactive carbon or 14C. Okay, by using radioactive carbon or 14C. Okay, and Melvin Kelvin was working. The Melvin Kelvin was working in a algal photosynthesis, in algal photosynthesis to reduce this cycle. Okay, so Melvin Kelvin reduced this cycle. He used radioactive technique to reduce this cycle, and he used algal photosynthesis to reduce this cycle. Okay, this is very simple. So we have to understand why it is called as C3 cycle. This is called C3 cycle because the first stable product here is a 3 carbon compound. Okay, and the 3 carbon compound, which is a stable product in this, is triose no sorry phosphoglyceric acid. Triose phosphate is formed after photosynthesis. We will be talking about it later. The first 3 carbon compound which is stable here is phosphoglyceric acid. Okay, phosphoglyceric acid. Okay, or more perfectly it is called as 3-phosphoglyceric acid. Okay, and a interesting thing here is there are some plants in which the first stable product is a 4 carbon compound that is known as C4 cycle. In the cycle that happening in plants, is known as C4 cycle. In those plants also C3 cycle is occurring. Okay, what I mean to say is, in those plants where C4 cycle is working, C3 cycle must occur in those plants also. So basically it is an important conclusion that this C3 cycle, this C3 cycle happens in both C3 and C4 plants. The C3 cycle must occur in some stage in any plant to survive, to survive the plant. The plant must must be performing the C3 cycle. Okay, and in the mesophyll cells of C3 plants, this C3 cycle is this C3 cycle takes place. Okay, clear? So far what we have studied. So Kelvin cycle, Melvin Kelvin, radioactive carbon, algal photosynthesis, C3 cycle, because the first stable product is a 3 carbon compound which is 3 phosphoglyceric acid, and 3 C3 cycle happens both in 3 carbon compound, both in 3 C3 plants and C4 plants. C3 cycle must be happening in any plant. Okay, otherwise the plant cannot survive. So we will be studying about stages of Kelvin cycle. Okay, stages of Kelvin cycle or C3 cycle. So the first stage of the Kelvin cycle is carboxylation. In carboxylation, the CO2 is added with a compound which is known as RUBP. This is ribulose bisphosphate. Okay, and the enzyme here is Rubisco, and the product here formed is 3-PGA. I told you that 3-PGA is a 3 carbon compound. Okay, and this RUBP here is a 5 carbon compound, and this is a carbon dioxide, and this is 3-PGA, and this is 3 carbon compound. And if we balance the reaction, it happens to be 2 into 3-PGA, 2 molecules of 3-PGA, 3 phosphoglyceric acid. So 3 into 2, 6 carbon. So 3 into 2, 6 carbon here, and 5 plus 1, 6 carbon here. The equation is balanced. And note an interesting thing here, the first carbon compound is a 3 carbon compound. So scientists in earlier age thought that, I am noting here a small note, that at first scientists thought the primary acceptor of CO2 is 2 carbon compound, because the product here is a 3 carbon compound. The first scientist thought in the earlier age that the acceptor here is a 2 carbon compound, but later it was disapproved because Kelvin found RUBP, which is a 5 carbon compound, as CO2 acceptor, here. Okay, fine. So the enzyme, the name of the enzyme here is Rubisco. Okay, Rubisco, ribulose bisphosphate carboxylase oxygenase. Ribulose bisphosphate carboxylase oxygenase. Okay, if you don't find the spelling here, Google the enzyme Rubisco, you will be finding the full spelling there. So why this is called Rubisco? Because this enzyme has carboxylase, that is ability to bind with carbon. The enzyme has carboxylase activity also, and the enzyme has oxygenase activity also. What is oxygenase activity? Oxygenase activity is ability to bind with oxygen also. So this Rubisco enzyme has both the abilities to bind with oxygen and to bind with carbon dioxide. But the tendency to bind with carbon dioxide here is more. Why? Because our atmosphere contains more oxygen and very less carbon dioxide, and the enzyme binds here mostly with carbon dioxide. So the affinity to carbon dioxide to this enzyme is very high. Okay, and the second step here is, first step carboxylation, the second step is reduction. So in the product we have found 2 into 3 PGA, 2 molecules of 3 PGA, 3 phosphoglyceric acid. So here 3 phosphoglyceric acid is converted into 2 molecules of triose phosphate. If you have read the glycolysis very carefully, you will be understanding this. In glycolysis, this triose phosphate converts into 3 phosphoglyceric acid, but in case of photosynthesis, this phosphoglyceric acid converts into triose phosphate. So this reduction is sometimes called reverse of glycolysis also. Okay, and in this step, listen this very carefully. In this step, for every CO2 fixed, for every CO2 fixed, 2 ATP is converted to 2 ADP and 2 Na and 2 NADPH is converted into 2 NADP plus here. So every CO2 fixed, for every CO2 fixed, 2 ATP is converted to 2 ADP and 2 NADPH is converted to NADP plus in case of reduction. This is happens in reduction only. Okay, this thing. Okay, so this is one molecule of CO2 is fixed here. So here 2 ATP and 2 ADP and 2 NADP plus is equal to 2 NADP plus. Okay, 2 NADP plus. Okay, this is a very important thing, and this is reverse of glycolysis. Okay, and if you have a good memory, I have taught this that this C3 cycle is basically a biosynthetic phase, and biosynthetic phase means biosynthetic phase means we are using the products of light reactions here. So what are the products of light reactions? Listen, look at this, very very interesting. This is the product of light reaction, ATP, and this is also product of a light reaction, that is NADPH. And this both thing is being used here. Look, this is the first step where the products of light reaction is used. So there the product of light reaction, that is ATP, is converted to 2-ADP, and the product of light reaction, that is 2-NADPH, is converted to 2-NADP plus. Okay, so it's very clear, this is okay. This is reduction. The third step here is regeneration. What happens in regeneration step? In regeneration step, our previous product of the second reaction, that's the reduction, our previous product reduction was okay. And our previous product was here 2 into 2 triose phosphate molecule. Okay, so 2 triose phosphate, and triose phosphate is a 3 carbon compound, so basically 2 into 3 carbon compound is 6 carbon. 2 triose phosphate, it converted into a RUBP, which is the acceptor, which is a CO2 acceptor, which is CO2 acceptor, RUBP, and RUBP is a 5 carbon compound. So here 2 into 3 carbon, this is 6 carbon, and here 1 carbon is used in making carbohydrate. Okay, this 1 carbon here used in making here carbohydrate. 1 carbon in, listen, this is also very important. I am using 1 carbon for making carbohydrate for 1 molecule of CO2 fixed. Okay, and in this step also, for 1 molecule of CO2 fixed, for 1 molecule of CO2 fixed, 1 for 1 molecule of CO2 fixed, 1 ATP is converted into 1 ADP in regeneration. So this is very clear that for 1 molecule of CO2 fixed, 1 carbon is used in making carbohydrate, 1 carbon is making sorry 1 carbon is used in making carbohydrate for 1 molecule of CO2 used, and for 1 molecule of CO2 fixed, 1 molecule of CO2 fixed, 1 ATP is converted to ADP in case of regeneration. So here ATP is converted to ADP here, 1 ATP to 1 ADP. This regeneration. Okay, so I am summarizing this whole Kelvin cycle in a cyclic manner in the next slide. So what is happening in the first? So what is happening in the first? The first RUBP which is a 5 carbon compound, is combining with CO2 and giving rise to into 2 into 3 PGA. The 3 PGA here is a 3 carbon compound, so 5 carbon, 1 carbon. This is 1 CO2, okay. This is 1 CO2. This is very important, 1 CO2 here entering. 2 into 3 PGA, that is 3 carbon. And the enzyme here is Rubisco. And the 3 PGA here, and the 3 PGA is converting into triose phosphate. And while converting into triose phosphate, 2 NADPH plus 2 ATP is converting into 2 NADH, 2 NADP plus 2 ADP. Why? Because I have told you that for 1 CO2 fixed, in case of reduction process, this is reduction here, and the first step is carboxylation. In this reduction process, for 1 CO2 fixed, 2 NADPH plus 2 ATP is converted into 2 NAD plus 2 ADP, and this 2 NADPH plus 2 ATP is a product of light reaction, if you remember, product of light reaction, okay. So this is converted into triose phosphate. Triose phosphate is 3 carbon compound, so 2 into triose phosphate. And this triose phosphate, this is converted is again into in the step named regeneration to IUPP, and here 1 carbon is used in synthesizing carbohydrates, okay. So 1 carbon is used in synthesizing carbohydrate here for each CO2 molecule fixed. This is also important, and I have told you in the previous slide that for each CO2 molecule fixed, here also 1 ATP is converted to 1 ADP for this regeneration process, okay. This is 1 ATP, this is 1 ATP, regeneration process. This is very clear. So let's summarize. So how many ATP are used? So how many, and a important thing I forgot to tell you, that this RUBP is a 5 carbon ketose sugar. This RUBP is a 5 carbon ketose sugar. Look, this point I am telling you here is given in NCERT. Look very carefully in NCERT, that RUBP is a 5 carbon ketone sugar is given in NCERT. Ketose sugar is very important. I forgot to tell you this point. This is a very important point. This is 5 carbon ketose sugar is RUBP, okay. So we are summarizing the ATP calculation and ADPH calculation in the next slide. Okay. So what is the ATP? So I am writing for 1 CO2 fixed, what are the things happened for 1 CO2 fixed? 2 ATP in the process of reduction is used, and 1 ATP in the process of regeneration is used. So a subtotal of 3 ATP is used for 1 CO2 fixed. And how many NADP is fixed? How many NADP is used? 2 NADPH is used in the process of reduction. So what is the total NADPH? The total NADPH comes to be 2 NADPH NADPH in reduction and 3 ATP for 1 CO2 fixed. And also 1 carbon is used to synthesize 1 carbon is used to synthesize carbohydrate, that is glucose, fructose, sucrose, whatever it be, carbohydrates. Okay, to synthesize carbohydrate for 1 CO2 fixed. So tell me, this is a simple calculation. So for 1 CO2 fixed, 3 ATP used. For 1 CO2 fixed, 3 NADP is used, and 1 carbon is used in to synthesize carbohydrate. Okay, so glucose, suppose glucose has 6 carbon. Okay, that means the plant has to use one molecule. So plant is forming one molecule of glucose. So how many ATP and ADP he will use? The plant will use. So for synthesizing one molecule of glucose, which is a 6 carbon compound, the plant needs to use 6 carbons. 6 carbons means the plant has to fix 6 CO2 fixed, 6 CO2 fixing, and 6 CO2 fixing simply means 6 into 3 ATP, because in 1 CO2 fixing 3 ATP is used, and for 3 CO2 fixing 6 into 3 ATP used, and 6 into 2 NADPH is used. Why? Because 2 NADPH used is in synthesizing in using 1 carbon to synthesize carbohydrate or fixing 1 CO2 in the Kelvin cycle. So 6 into 2 NADP is used. I am repeating this line again, that 3 ATP is used to use 1 carbon to form carbohydrate. Okay, 3 ATP is used to pump 1 carbon into carbohydrate. So to pump 6 carbon into carbohydrate, we have to use 6 into 3 ATP. Okay, 6 into 3 ATP to pump 6 carbons, and to pump 1 carbon into carbohydrate, we have to use 2 NADPH. I have discussed already here and here. So we have to pump 6 into 2 NADPH to pump 6 carbons into carbohydrate, or that is to form 1 molecule of glucose. The question may be asked that suppose 3 molecules are used in forming glucose, or 12 molecules is forming glucose. You have to just multiply it accordingly. This is the concept, basically concept here. You have to understand the concept, and the calculations will be done accordingly with the carbon number. So we have to glucose, we have multiplying in it with 6, and you have to suppose this is a 12 carbon compound, you will be multiplying this number with 12 here. Okay, so this is simple. So this is 18 ATP plus 12 NADPH basically. Okay, simple. 18 ATP and 12 NADPH for forming 1 molecule of glucose in Kelvin cycle. Okay, this is all about Kelvin cycle. In the next slide, we'll be talking about C4 cycle. Okay, C4 cycle. So why this is C4 cycle? I have told you previously, I have given a basic concept. It is called C4 cycle because the first stable compound here is a C4 acid. I am writing it here. The first stable compound here is a 4 carbon is a 4 carbon acid 4 carbon acid, and the name of the acid is oxaloacetic acid, OAA, oxaloacetic acid, oxaloacetic acid, OAA, 4 carbon acid. So C4 cycle, and in which plant the C4 cycle is happening is known as C4 plants, and in C4 plants this C3 cycle is also happening, as I told you previously, is also happening. Okay, so what is the specialization of C4 cycle? The specialization of the C4 cycle has large cell around vascular bundles which is known as bundle sheath cells. So C4 plants have large cell around vascular bundle which is known as bundle sheath cells. Okay, it is a basic it is also in the plant anatomy. While you will be reading plant anatomy, you will be learning about this also. So large cell are around vascular bundle which is known as bundle sheath cell, and leaves with such cell is known as leaves with such cell and if leaves with such cell are present in a plant, we call that the plant has the a Cran's anatomy. Okay, so while leaves with such cell, which cells? Leaves with such bundle sheath cells. Okay, what is bundle sheath cell? Which is large cell around vascular bundle. So leaves with such cells are called Cran's anatomy, or basically Cran's anatomy is a type of anatomy where leaves with such cells are present. Okay, it is the full definition what I am writing. Cran's anatomy is an anatomy where leaves with bundle sheath cells, which is a large cell around vascular bundle, are present. So Cran's anatomy is an anatomy where leaves with bundle sheath cell or large cell around vascular bundle are present. This is the basic definition of Cran's anatomy. And there are some also specialization about this vascular bundle cell. This vascular bundle sheath cell. What time right? This bundle sheath cell. What is the specialization? The bundle sheath cells forms several layers around the vascular bundle. Okay, it has large amount of chloroplast. It has a large amount of chloroplast and it is impervious, very large amount of chloroplast. Okay, and it has no intercellular space, no intercellular space. So because there is no intercellular space, so it is impervious to atmospheric gases. So it is a gas proof chamber, the bundled cell. So because there is no intercellular space, no gas can enter out from outside into the bundled cell. Okay, and the examples of such C4 plant in which the bundled cell are present are bundled cell are present in where craniotomy is also present, that is maize and sorghum, sorghum, sorghum. Okay, and have I forgot something to tell you? Let me check. Bundle cell forms several layers of the vascular bundle. Okay. So this crans means wreath. Wreath means a ring-like structure. Wreath means a wreath. This is round thing, wreath. And you know the lips are there, and around the vascular bundle there are bundle seed cells, and these bundle seed cells forms a large thick layers around the vascular bundle, and this anatomy is called as Grant's anatomy, and this is the specialization of the C4 plant which is present in the plants. Okay, in Grant's anatomy various many chloroplasts are present in the bundle cell, and in bundle cell no intercellular spaces are present. So again I am remembering you, in C4 plant there is vascular bundle, and outside vascular bundle there are layers there is layers of bundle cell which forms a wreath like structure which is known as Cran's anatomy, and these bundle cell forms several layers around the vascular bundle, and it has large amount of chloroplast, and this bundle cell also has no intercellular spaces, so no gas can enter from the atmosphere to this bundle cells. Okay, and the examples of some such C4 plants which is given in NCERT book is maize and sorghum. This is very clear. Okay, so in the next slide we will be talking about the C4 cycle. Okay, clearly clear. C4 cycle. So basically what happens? Wait a minute. Okay, So this basically is mesophyll cell, and this is here is bundle, and this is basically here is bundle sheath cells. Okay, and in this mesophyll cell, in the atmosphere there is CO2. CO2 is fixed with H2O from the atmosphere, and it forms bicarbonate ion here, HCO3 minus ion here, and this HCO3 minus ion here adds with phosphoenol pyruvate to form oxaloacetic acid. Okay, this is a 4 carbon compound, so this is known as C4 cycle, and this has 1 carbon HCO3 minus, and this PPS3 obviously 3 carbon. So the first electron acceptor, so the electron acceptor here is a PP or phosphoenol pyruvate. Electron acceptor here is PP, not electron, not elect sorry, not electrons. Sorry. This is CO2 acceptor. This is CO2 acceptor here is basically is phosphoenol pyruvate. Okay, not only CO2, this is primary CO2 acceptor here. Primary CO2 acceptor here is phosphoenol pyruvate, and remember, in case of C3 cycle, the primary CO2 acceptor was RUBP, ribulose bisphosphate, which is also a 3 carbon compound, and in case of C4 cycle, the primary CO2 acceptor here is phosphoenol pyruvate, which is a 3 carbon compound, and the first stable product is a oxaloacetic acid. This oxaloacetic acid is the first stable product here, oxaloacetic acid, and this oxaloacetic acid gets converted into malic acid or aspartic acid in the mesophyll cell, and after converting into malic acid or aspartic acid, which is also a C4 acid, it is transported into the bundle sheath cell, okay, as a C4 acid, and here decarboxylation occurs. Here and here decarboxylation occurs. Okay, so on decarboxylation, here one CO2 gets popped out, and a C3 acid is formed, because one CO2 means 1 carbon and C3 acid means 3 carbon, and this is C4 acid. From C4 acid 1 CO2 and C3 acid is formed, and this C3 acid is again, this is, let me think, this is transport, and this C3 acid is again transported into mesophyll cell, transported in mesophyll cells, and after transporting it, it is the regeneration of the phosphoenol pyruvate occurs. What is the regeneration of phosphoenol pyruvate? The regeneration of the phosphoenol pyruvate from the C3 acid occurs in this step. And remember, this regeneration process for fixing for fixing also one carbon molecule here requires a net of 1 ATP to 2 ATP to 2 ADP here. Very clear. Okay. So let me come to the bundle sheet cell. So in the bundle sheet cell, here is CO2 is popped out, and this CO2 here goes to the Kelvin cycle. Kelvin cycle here. So basically the Kelvin cycle here occurring in bonded cell in case of C4 plant. Okay, I will be writing there in a separate page. So remember the Kelvin cycle occurring in bonded cell in case of C4 plant, and the Kelvin cycle occurs mesophyll cell in case of C3 plant. Okay, but Kelvin cycle or C3 cycle is necessary here. Is very necessary. You have to understand it. So let's come to the ATP calculation in case of C4 plants. So let's calculate for 1 CO2 fixed. Remember in the C4 plant C3 cycle is also happening in the bonded cell. So for 1 CO2 fixed there is a net 3 ATP and 2 NADPH used in case of C3 cycle. Already it is used in the C3 cycle, and a extra of 2 ATP is used. This is of C3 cycle, and a extra of 2 ATP used for regeneration of the acid. Regeneration of the phosphoenol pyruvate from the C3 acid here. Regeneration of phosphoenol pyruvate here. Okay. Oh, sorry. I have forgot to mention the name of the enzyme here. The enzyme here which is responsible for the fixation of the carbon dioxide or the carbonate ion here is PEPCO. PEPCase. 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 Okay. The name of the PEPCase is phosphoenol pyruvate carboxylase. PEPCase. Phosphoenol pyruvate carboxylase. The enzyme of the name here is PEPCase. This is important. Phosphoenol pyruvate carboxylase. And what is the enzyme in the C3 cycle, which is also happening in this bonded cell? Enzyme in the what is the CO2 fixation enzyme in the C3 cycle or the Kelvin cycle? Remember, I have taught in the previous slides. That is the Rubisco, ribulose bisphosphate carboxylase oxygenase, and in case of CO2, in case of C4 plants, it is PEPCase, phosphoenol pyruvate carboxylase, and in case of C3 plants, it is Rubisco or ribulose bisphosphate carboxylase oxygenase. Okay. So don't worry, I'll be writing this point in this separate points also. So let us come to the ATP or net calculations here. So here basically in the C3 cycle 3 ATP and 2 NADPH, and for regeneration 2 ATP used for 1 carbon dioxide molecule fixed. So how many? 5 ATP is used and 2 NADPH is used for fixing 1 molecule of carbon dioxide here. Okay, this is very clear. So for forming glucose, that is a 6 carbon compound, remember the previous calculations, 5 ATP into 6 is required plus 6 into 2 NADPH is required. So for formation of a glucose molecule here, 1 glucose molecule here, 30 ATP molecules are required plus 12 NADPH molecule is required. So this requirement is for C4 plant, and if you remember, in case of C3 plant, one glucose molecule required 18 ATP plus 12 NADPH. So compare this with C4 plant. In case of C4 plant, 12 ATP more used than C3 plant for forming one glucose molecule. Okay, so this is 12 ATP plus 12 NADPH plus 18 ATP for one glucose molecule, and here is 30 ATP plus 12 NADPH for one glucose molecule. So very clear. Okay, very clear. Okay, so let us write down some important things for C4 plants and C3 plants. What are the things? The things are, let us take as C3 plant and C4 plant, where the things we forget are, in case of C3 plants, in mesophyll cell, in case of C3 plant, in mesophyll cell, the Kelvin cycle is occurring. Okay, but in case of C4 plants, in bundle sheath cell, the Kelvin cycle is occurring. Okay, and here primary CO2 acceptor, and here the primary CO2 acceptor is RUBP, ribulose bisphosphate, and here the primary CO2 acceptor is phosphoenol pyruvate. Okay, and here the first stable product is a C4 acid, which is oxaloacetic acid. This is C3 acid. This is C3 cycle, this is C3 acid, which is 3 PGA, 3 phosphoglyceric acid. First stable product, and it is a C4 acid here, which happens to be oxaloacetic acid. This is first stable product here. Okay, and if you have been asked then that in the mesophyll cells, in case of C3 plants, which enzymes are found in mesophyll cell, you will be answering that in case of C3 plants, the mesophyll cells has the CO2 fixing enzyme or Rubisco. Okay, but this Rubisco enzyme is present in bundle cell, because the Kelvin cycle is happening in the bonded cell in case of C4 plants. I am writing here, if we talking about Rubisco here, it is in case of C3 plants, it is situated in mesophyll cell because the C3 cycle is happening there, but this Rubisco enzyme is situated in bundle sheath cell, because the in the banded cell the C3 cycle is happening in case of C4 plants. Yeah, I hope you have understood it. If you haven't understood it, rewind the video and watch it again. This is a very important comparison between the C3 and the C4 plants. You have must remember it. It is very important. And in case of C3 plant, for one glucose molecule, there is a total of 18 ATP plus 12 NADPH is required. And in case of C4 plant, for one glucose molecule 30 ATP plus 12 NADPH is required. Okay, very clear, 30 ATP plus 12 NADPH. Okay, and we'll be concluding our lecture after understanding an important point. So we have to understand why do we need C4 cycle while we have C3 cycle. Why do we need C4 cycle while we have C3 cycle? And ask yourself this question. The C3 plants, there is also CO2 fixation is occurring, and in C4 plants also CO2 fixation is occurring. But in case of C4 plants, the ATP utilization is much more than the C3 plants. So why do you need the C4 plant to exist? Okay. So if you remember, the primary carbon fixation enzyme was, in case of Kelvin cycle, was Rubisco. And if you remember, this Rubisco has both oxygenase and carboxylase activity. This Rubisco enzyme has both oxygenase and carboxylase activity. Okay, so that means Rubisco has both oxygenase and carboxylase activity. That means Rubisco can bind with both O2 and CO2 depending upon the relative concentration between them and various other factors. Okay, so this Rubisco enzyme can also bind. So what I'm trying to say that this Rubisco can bind with O2 also, and Rubisco can bind with CO2 also. But you have to remember the affinity to bind with CO2 is more. Why? Because this CO2, if you remember, the relative abundance of CO2 in atmosphere is 0.04% and O2 is about 20%. So O2 is much more. So O2 is present in much more amount than CO2 in the atmosphere, and O2 is 20% and CO2 is only 0.04%. But it is a carbon dioxide fixing enzyme, and it helps in photosynthesis. So we have to understand the affinity to bind with CO2 here is more, because it is in the most of the cases it's bind with CO2. It binds with CO2, but the enzyme can bind also with O2 and also with CO2. So the problem happens. The problem starts when the relative concentration of O2 is much more in surroundings, the Rubisco enzyme binds with O2 and the Rubisco enzyme binds with O2. So you have understood that. When the relative concentration of O2 is much more in surrounding, the Rubisco enzyme binds with O2, and on binding with O2, this on binding with O2, eraser, on binding with O2, the RUBP, on binding with O2, the RUBP, which is the substrate of C3 cycle, loses CO2 molecule, and this harmful process is known as photorespiration. So while the concentration of O2 is high in the surrounding, this Rubisco enzyme binds with the RUBP with the help of Rubisco enzyme. The RUBP binds with CO2. Binds with O2. This RUBP binds with O2 with the help of Rubisco enzyme when the surrounding O2 concentration is high, and loses CO2 molecule. In the C3 cycle, if you remember, the CO2 molecule is getting added but when the relative concentration of O2 is high, this RUBP binds with O2 and loses CO2 molecule, and this harmful process is known as photorespiration. So basically what happens, after binding with O2, the RUBP loses CO2 molecule and forget glucose. Glucose is not forming. Loses CO2 molecule and glucose is not forming. The carbons which are present in the carbons which are present in the plants only, they are also getting away from the plant. So basically, carbon of the plants gets reduced, so the photosynthesis here the photosynthesis get reduced here. The photosynthesis rate get reduced in case of C3 plants, okay, because the carbon of the plants is get reduced. And in case of photorespiration, in which the binding of O2 occurs with RUBP, And in case of photorespiration, ATP is also utilized. So the photorespiration is absolutely harm process with the binding of IUPAC with O2, that is photorespiration. Photorespiration is absolutely a harmful process for plants because firstly it does not accept electron, so no profit. Sorry, it does not accept carbon dioxide, so there is no profit. Rather, it loses carbon dioxide, it loses carbon, and it utilizes ATP. It does not benefits, but it harms. So this photorespiration process is a absolutely harm process. This is a very harmful process. Okay. It's a very harmful process. So to minimize this process, to minimize this harmful process, plant has developed C4 cycle here. So to minimize the harmful photo respiration in which the yield of the C3 plants gets reduced. The yield of the C3 plant get reduced. Plants have developed C4 cycle. Okay. And if you remember, I have taught you that C4 plants have Kranz anatomy. Remember, bundle sheath cell, no intercellular space. So, because there is no intercellular space, no gases means no O2 from outside bundle sheath cell. No O2 from outside bundle sheath cell is allowed to enter in the bundle sheath cell. So in the bundle sheath in the bundle sheath cell, so in the bundle sheath cell, the CO2 concentration is very high because decarboxylation of C4 acid occurs here. Remember, this is a very important point I have taught you previously. Look at the previous slides. Look, I am taking you to the previous slide. Look here, this is bundle sheath cell. This is bundle sheath cell, this is bundle sheath cell, and this bundle sheath cell have no intercellular space. So there is no O2 from the outside. So the relative concentration is O2 is null in the bundle sheath cell. So because of the null O2 concentration here, Look this CO2 here is decarboxylated in the bundle steel cell. The relative concentration of CO2 is very high in this bundle steel cell. So because of this high relative concentration, the carboxylase function of Rubisco here gets maximized. Let me write this thing in the next slide. You have understood what I am trying to say, that the CO2 is decarboxylated in the bonded cell, and here is no intercellular space, so no O2 is coming from the outside. So the carboxylase function of the Rubisco here is gets maximized, and the oxygenase function here gets minimized. Okay, so here I'm writing that the carboxylase function here gets maximized for C4 plants, and oxygenase, that is to addition with oxygen with IUPAC, oxygenase function gets minimized. So in case of C4 plants, so the percentage yield of the C4 plants, or the photosynthesizing efficiency of the C4 plants, are more than the C3 plants. Okay, so the photosynthetic ability of the C4 plant is more than the C3 plant. Okay, so this is the reason. Of C4 plants this is the reason of C4 plant being more photosynthetically active. And we have also understood that in C4 plants there is no photorespiration. I have told you already, in C4 plant there is no photorespiration because there is no oxygen activity in the Rubisco enzyme, so there is no photorespiration. So C4 plants have maximum photosynthetic ability, and the percentage yield of the C4 plants is more than that of C3 plants. Okay, so we will be discussing the photorespiration in our next slide, in our next lecture, and this is the end for today. So thank you, students. Please subscribe to my channel for the next updates. Please subscribe. You see my video and don't subscribe my channel, it is very bad, students. Please subscribe my channel. My channel is very good for NEET preparation. I have concised the NCERT book with some outside data, and to understand you the basic concepts, and my channel and what I have presented in front of you in this chapter is absolutely sufficient for NEET. Okay, so please guys, subscribe to my channel. Namaste, Jai Hind.